Good afternoon, everybody. This is Ed Helmig from Arcadis, and we have a pretty exciting lunch hour presentation for you. In fact, we're going to start with a little food. We'll get to that in a minute. But first, I wanted to get a show of hands. Uh, you can find your hand button down at the bottom of your screen. How many attendees here are from industry, meaning manufacturing of pharmaceuticals, chemicals, or other type products? Quick show of hands. Okay. How many people are from government? Not seeing too many. How many people are from consulting? All right. My screen mustn't be working. Uh, Nobody's identifying themselves. That's okay. Uh, this is primarily geared towards people in consulting and industry. And Dave and I are going to walk you through a presentation today. You can go to the next slide, Dave. So I promised we would start this with lunch, and I figured everybody would like a nice frothy cold beer. But how would you like that beer dosed with hormones and a little bit of selective serotonin reuptake inhibitor like Prozac and a plate of fish with the drugs listed there? Of course, nobody wants that, right? And that's our job as environmental engineers, is to prevent that kind of thing. So I've got on the left-hand side of the slide there some actual case histories, just as quick bullets. But I'm only going to talk about one of them. And this was a hormone that was found in pig feed and soft drinks and triggered a criminal investigation, a fine to the company, and damage to its reputation. So this was a big issue. Many in the pharmaceutical industry are aware of it. It also turns out that the factory was upstream of the intake of a brewery. So you can imagine the enormous risk potential of an operation like this. So this is real stuff, and it's very important. You can go to the next slide. So this chart is to let everybody know where this stuff is coming from. And so you can see wastewater influent, which rarely gets discharged directly, at least in first and many second world countries, but you can see the effluent from treatment plants. And most treatment plants are not designed nor capable of removing many of the hundreds to thousands of pharmaceutical active ingredients that are found in wastewater. And then you can see surface water, soil, drinking water, and groundwater levels by comparison. So wastewater treatment plant effluents are a big source. You can go to the next slide. So I want to pull a common thread today, and that thread is as follows. We're going to start by talking about how do you define the scope and magnitude of your problem, the risk, okay? And then we're going to go into what the next critical step is in a project or solving this kind of problem, which is to spend time understanding the manufacturing process. You need to understand the waste stream characteristics and volumes. You need to learn where these waste streams are coming from, how they're generated, and how they might be segregated or treated at source, because end of pipe treatment tends to be expensive. And you need to develop a sampling strategy that's going to complement your predicted environmental concentration estimates and help you validate those numbers. And then you're going to need to understand what you need to do to treat the APIs. And that's going to have a lot to do with what's in the wastewater in terms of a molecule and the matrix that it's in, and then the technologies that are available to you and the ones that really work. And that leads to treatability, which is where we get to theory versus reality. And let me tell you, you're going to find out, as we have, that a lot of stuff that you read in the literature just doesn't work or isn't scalable. And we'll talk about pre-treatment. Sometimes these organics are present at very low levels. So in order to remove them, you need to get all the other junk out first. And then Dave will talk to you about scale-up decisions, that can be made after treatability. We'll talk a little bit about energy and process safety. So that's the common thread that we're going to pull throughout this presentation. So you can go to the next slide. So let's talk about the categories of APIs that we have out there. So Dave and I tend to divide them into three different categories. We have endocrine disrupting compounds, hormones like 17-beta, ethanol, estradiol, trimegesterone, those type compounds that actually have an endocrine effect on fish and humans and other receptors. Oral contraceptives is a subclass under there. Postmenopausal hormone replacement drugs, HRTs, is another subclass. Then we have toxic compounds, compounds that will actually kill cells or exhibit a toxic effect. And then we have antibiotic and antimicrobial agents, which are a special class of compounds that have some interesting effects. And we'll talk about those a little bit. So there they are in three nice little circles. And the circles overlap because they're, compounds that can be an endocrine effect can also be have some toxicity. So the point here is it's not just the compounds, it's measuring the effects of the APIs, the active pharmaceutical ingredients, that is equally as important and can be very difficult and time consuming. So anybody who's ever heard of the yeast estrogen receptor assay, which is used to measure endocrine effects, knows that it's a complicated test. And we've done it and used it on projects. And it's, if you talk to the people who develop it, it's not an easy test to do. We go to the next slide. So let's focus a little bit on antibiotics for a minute. So these are very important drugs. All of us here, I think at one time or another, have probably taken antibiotics, and they've saved millions of lives. And it's not just penicillin. Penicillin was the beginning of antibiotics in the 20s and 30s, and there's the class of antibiotics out there now is huge. So they're designed to kill certain kinds of cells, and they have a toxicity threshold. So you need to think about that when you're thinking about biological treatment, right? The dose makes the poison. So not, antibiotics, there are different concentrations that are going to cause a problem. And that's reflected in your predicted no effect concentrations for the specific compound, and also in toxicity and inhibition studies that you would do for your biomass. And then, bacteria are clever. They evolve quickly. They can develop resistance to antibiotics, rendering them ineffective. And that's one of the things that makes antibiotics a special class of compounds, because bacteria can acquire antibiotic resistance in the environment. And if those bacteria get into your body, and let's say it's streptococcus, and you go for treatment, and they give you the preferred antibiotic for streptococcus and it doesn't work, it's because you probably have a superbug or a antibiotic resistant organism inside of you. So as I said, this antibiotic class is huge. This is homework here for you, but what we've done is we've put them in nice boxes there. There's 12 boxes, starting with the beta-lactam antibiotics. I think everybody's probably taken amoxicillin at one time or another. So in each box, we describe the bacteria based on how it works. So the beta-lactams inhibit bacterial cell wall synthesis, and then you go down through the tetracyclines and macrolides and stuff. So this is homework for you to study. This is a very interesting area to look into. Pathways to the environment. This is pretty simple. Let me go from the general population there on the left. These are widely dispersed, low concentration sources. And then on the far right, you see industry, and these are point sources that are highly concentrated. You can think of a tablet. People taking a tablet, it gets maybe 70, 80% metabolized, and the metabolic process plus the original API end up being excreted and and put it into the environment. In industry, in a tabulating plant, you're typically making 1,500 kilogram batches. And if you lose 1 or 2%, that's a lot of material going down the drain, and you're doing batches back to back to back with cleaning in between each batch. So now let's talk for a minute about how does, with regard to antibiotics, how does antibiotic resistance occur? So let's take a treatment plant, for example. You have an antibiotic going to a treatment plant. It's loaded with bacteria, and those bacteria, some of them are going to survive that antibiotic. So you have a selection effect. They're going to develop enzyme systems to degrade that antibiotic, and they're going to incorporate that into plasmids, okay, and they're going to be able to pass that along to other cells through a process called horizontal gene transfer. Now, horizontal gene transfer only occurs among prokaryotic cells. That's bacteria, protozoa, and archaea. It does not generally occur between eukaryotic cells, which are your mammalian cells, the cells of our skin, for example, animals, fungi, and paramecium. So basically, plasmids are like cassettes that are being thrown out the window when a cell dies, and other cells pick up those plasmids, and when they're under stress, they can pick those cassettes up and play them, and what they play is um, the the information required to defeat an antibiotic, and those can get passed from generation to generation. So a plasmid is just a small circular strand of DNA, and it's separate from chromosomal DNA. So we can go on to the next one. So, we're going to talk about estimates and measurements here. And this is a tedious, high precision task. When we're talking about APIs in the environment, you're talking about, like I said, 1,500 kilogram batches, where the PNEC, predicted no effect concentration, might be a tenth of a nanogram per liter. So you can be talking about 12 orders of magnitude difference. And Dave's going to talk to you about how you go about doing those estimates.
Thanks, Ed. So really, when you're embarking on these estimates, and they at their core are a mass balance effort. But really, what you need to do is start by understanding the production process. Really mapping it out, understanding where things are moving throughout the process, where there are losses, and then how it goes into the sewer, and what happens downstream of the production process. Um, Understanding production schedules is critical for this, both from developing the estimate, but also when you are doing information sampling. Um, So this is an example of a typical fermentation-based production process. The big thing to note here is really understanding where the active ingredient first is expressed. And that's usually at some point in the fermentation process, which is typically a multi-day process. But really, the big areas of concern are going to be downstream of fermentation, when it's being extracted, purified, and concentrated up. Those are where you're going to see your biggest losses. Um, that's not to say you shouldn't or you should overlook the upstream processes, because there are events that can happen that could lead to a contribution from up there. So as I said, this is really a mass balance effort, going back to first principles there, and it's really understanding the process to come up with a reasonable estimate. So got a couple of examples here, and really what we like to do is take that complex process map and distill it down to key blocks, create a block flow diagram, and identify the streams of interest that are have the active ingredients in it. So in this case, we've got a bunch of different color codings um, for different destinations for those streams. And then also whether they are kind of a continuous or routine discharge versus an abnormal discharge or an end of campaign discharge. That all feeds into how you prepare the estimate. As I mentioned, some of the upstream, like the fermentation processes, there can be some unique events that really you don't learn about until you start talking to the production staff. And case in point is something where just due to the biological activity and growth in the fermenter, you might see some overflows that occur at random times. In the case of this compound, all the waste streams were aqueous. They had low solvent concentrations, so they were all generally directed to the sewer. Um, Other examples may have solvent recovery systems. So that could be another source that's outside your immediate production area that you need to keep track of. The other things is just understanding how they do the purification and extraction processes. They may have kind of a trail that is out of spec that they let go to drain at the beginning or end of the process. Um, and the thing to keep in mind with a lot of these is you're talking about fairly small percentages of the total batch size as a loss. But when you start penciling that out, it can actually play out to some relatively large numbers. I mean, a 0.2, 0.3% loss is still 15 kilograms. And that can be pretty significant when you are talking about nanogram per liter concentrations that you're concerned about. Another example of a process, this one here just illustrates that it had a lot of other waste streams going to other locations: dust collection, solvent recovery, solvent waste disposal. All those, like I mentioned, you need to keep track of those because those could end up in the wastewater stream, depending on how they're handled. Other thing to keep an eye on is how are off-spec batches handled. What do they do for that? Usually it's going to go to drain. That's an abnormal event that needs to be accounted for. And also that plays into sizing of any sort of treatment systems, is that could be a huge organic and nutrient load on a treatment plan. So once you've figured it out on a process level, you expand it out to the facility to do your overall estimating of your PEC and PNEC against the PNEC. And so really what you're looking at is what your production yield is, what your treatment works removal are. That's going to give you how much mass you see being discharged, and then from the production process to the treatment, and then out of the treatment. Once you get it coming out of the treatment, then you convert it to a concentration using the receiving water body flow or volume, and that's there's different guidelines for which flows or volumes you utilize. Typically, we see 7Q10 flows, and what you can do is do some sensitivity analysis to see the various impacts of what would happen if production yields changed or improved removal in a treatment works or different receiving water flows or volumes. And what you'll see is really the flow or the volume is probably going to be the biggest factor in this evaluation. So now that you've done those estimates, you may want to go through and sample to confirm because usually when you're estimating, it's a very conservative approach. So the big thing when you're doing that is to understand your production schedule and processes so that you are actually capturing a representative sample. Ideally, you're not just doing a composite sample and missing a major production discharge. Really, you should be looking to capture as much of um, that production profile as possible. So that may require setting up a composite sampler or collecting multiple samples over the course of several days to really understand the total discharge. Analytical methods, there's several out there. The real key point is that they only cover a limited number of active ingredients, maybe 120 or more, when there's 20,000 plus in the marketplace. So big things are understanding the capability of the method, understanding your APIs that you're looking for, and understanding the matrix. Those are all going to have impacts on the quality of your analysis. Ed?
Yeah, so let's talk a little bit about treatment here. This is, one of the most important tools or approaches that we have to reducing risk and preventing these APIs from getting out. Most of the projects we have done have been end of pipe treatment systems. We haven't just talked about them. We haven't just studied them and designed them. We've actually built them and put them into operation. And we've done a handful of at source treatment systems involving ultrasound and activated carbon and ozone and some other technologies. But keep in mind, lots of times in our business, you have to sell treatment to people. So you can't make the product if you can't treat the waste stream, and you need to get people to view it as a strategic asset that will enable the site to take on new products and processes, not just a operational liability. So we can go to the next slide there.
So when you're thinking about treatment, in the ideal scenario, you're involved at the very beginning when the production process is being designed and built. The reality is that's usually not the case. But if you are, the ideal scenario is you're designing it to reduce the losses to wastewater. And then you're looking at doing the PEC and PNEC evaluation to identify the key risks. But usually where we get involved is facilities already built and they're looking to reduce the losses to wastewater. So the first place to look is, all right, how is it being collected and discharged into the site sewers? Are there ways to segregate at or near source and capture key waste streams for targeted treatment or disposal? And then also just depending on the age of the facility, looking at the overall sewer infrastructure and repair it where possible to reduce the volume of additional water that you have to treat. But the goal is always looking to minimize the discharges as close to the source as possible. Once you've gone through that exercise, then it's really understanding, all right, what is required to treat, and that's going to be probably a multiple step process because your waste stream is going to have a lot of other stuff in it that needs to be removed before you can really get at the APIs of concern. So having proper equalization, pH control, solids, oil and grease removal, those are all critical components before you get into really looking at the APIs themselves and how you're going to break those apart or concentrate them or do what you want with them, which will happen in either chemical oxidation, maybe sorption, Sometimes biological treatment will do a pretty good job of it. And then in some cases, the best approach is really just to concentrate and incinerate. And then once you've gotten a treated effluent, evaluate the quality of it. What can be done with it? You probably produced a very high quality effluent. Are there ways to reuse it on site? So just a quick plug, always fractionate your influence COD, and understand what you have coming in, because there may be a lot of the compounds you're interested in are attached to particulates. And if you do some simple solids liquid separation through clarification, filtration, a dissolved air flotation system or the like, you're going to get 80+% of those compounds out, as well as taking a lot of other stuff out that will improve your downstream treatment. Then more often than not, you're probably going to end up in some sort of biological based treatment system. MBRs are a very good one for this because they have a more robust biomass, higher concentration mixed liquor, and then you have the advantage of that ultra filter on the back end that produces a very clean effluent that can then be treated with a chemical oxidant or activated carbon or something else. The one thing to keep in mind when you're dealing with a biological treatment system is the impacts on the biomass of the compounds. So this graph is just a typical respirogram showing an acclimated versus unacclimated biomass, the acclimated being the dash, the unacclimated being the solid line. The thing to keep in mind is most API production is done on a batch or campaign basis. So a biomass may acclimate to that particular compound or mix of compounds. Like I mentioned earlier, you have a lot of kind of synergistic effects that can happen. It may acclimate to that for a period, and then the production changes, and now have an unacclimated biomass again that has to acclimate to the new waste stream. So you're probably seeing a kind of this cycle repeat throughout the course of a year. So it's important to understand how the biomass will respond and ensure that you have a properly sized system that gives you that resilience. As I mentioned, MBRs are a very good system for handling these sort of waste streams. They're robust. They give a long sludge age that really helps with the biological degradation of some compounds. And then the effluent quality is very high. Once you've gotten through the primary and secondary treatment, then you're looking at tertiary to really get the last APIs out, get down to your target. So that could be additional filtration, some forms of mass transfer, ion exchange, or activated carbon, depending on the compounds you're dealing with. More often than not, you're probably going to be dealing with some sort of oxidation. And then depending on the waste, you may be dealing with a thermal system. Usually those are going to be on a smaller volume though. So event oxidation, that's really kind of a sledgehammer. It's going to either directly or indirectly oxidize the compounds. And so really understanding the chemical structure of each compound you're looking at is critical, to understand how it may be impacted by oxidation or by activated carbon or anything like that. There is always value in doing bench and pilot testing to confirm though. So this is just a quick summary of kind of the different or how each technology handles APIs, what the general capital range would be, and then kind of some of the operating considerations associated with each tertiary. So the Bottom line here is that each one has its pluses and minuses. It's really going to be subject to the waste stream, the compounds you're dealing with, and site specific. Ed?
Okay, yeah, this next slide is a very busy slide. I'm going to try to put it in a nutshell as quickly as possible for you. So this is arguably one of the more important slides in the deck and probably the most important project of my career. So remember in the beginning, I talked about the potential to have hormones and selective serotonin reuptake inhibitors in your beer. Well, this is the treatment plant that resolved that issue and made that risk disappear. So this was started in 2001 with the discovery that there was an issue and there was huge risk at the plant site with PEC to PNEC calculations. And it involved analytical method development, technology screening, bench and pilot scale testing, engineering design, equipment procurement, construction, commissioning, and then full scale performance verification. So I was involved in this project. I was the owner at the time at Wyeth, and I was involved in this project from the very inception until the very end when we turned it over to the plant and we had not only demonstrated unequivocally that this worked, it actually worked better than we had ever imagined. And I'll report, you probably can't see in the right hand box there the results, but the bottom line is what we built were a series of lift stations to capture all the wastewater. We ran through equalization with rotary drum screens designed to protect the membranes. So it's a membrane bioreactor system. We have biology tanks, hollow fiber membranes, a pressurized high performance ozone contact tank with a Venturi injection skid that can put anywhere from 30 to 300 milligram per liter ozone dose in, and then a degassing, a two-stage degassing system with a thermocatalytic destruct and an odor scrubber and so forth. So what's the bottom line on this? What did we validate this and prove unequivocally it could achieve? And so here's the answer in a nutshell. We got 99.94% destruction of the aggregate or total APIs in the wastewater, 1,000-fold reduction in estrogenicity from 637,100 nanograms per liter to 5.6 nanograms per liter of 17 beta ethanol estradiol equivalents. 98% COD removal, 99.9% BOD removal, 99% TSS removal, a non-toxic reusable effluent that was not only drinkable, but we reused half of it as makeup water and cooling towers. So when you know what you're doing and you do it right, this is what you can achieve. Go to the next slide. There's a paper on that project, by the way, that's written and published at WEF. So let's pull it all together. So again, a treatment plant's a strategic asset. You can see the one we just presented there as a huge strategic asset for that site, and it's still operating. So we've talked about how to define the magnitude and scope of your problem, understand the API's risk. That's the PEC and PNEC calculations. Understand the manufacturing process. You got to spend, depending on the size of the factory, a lot of time at the plant understanding what they're doing. Dave talked about the air emissions, the scrubber, the air emissions, the losses due to dust collectors. Those were scrubbers at the one plant we went to, and so the dust was going right back into the wastewater. And then treatability is critically important, especially if you're going to try newer novel technologies, which I wouldn't recommend if you're going to go to full scale. Try to stick with stuff that's been proven. So there we go. Thanks everybody for your time. Dave and I on the final slide have our contact information if you want to reach out to us. And I think we're going to open the floor to questions and answers.
Sure, absolutely. Please, if you all have any questions, go ahead and drop your questions right there in the chat module. Make sure that your chat option is changed to everyone. And go ahead and chat your questions. We'll give you just a few moments. And Dave and Ed, if you want to follow up on anything else, please take that opportunity to do so. I don't see any questions just yet in the chat.
While we're waiting for questions, Dave talked about analytical methods, and I thought of a question that people might ask, and that is, how do you know if an analytical method is any good? What's the criteria? So, in my experience, there are two criteria that I look at besides meeting the detection limit, because if it doesn't meet the required detection limit, it's no good. But the relative standard deviation and the percent recovery. So typical relative standard deviation should be in the 15 to 20% for a good method. The tighter, the better. And the percent recovery should be in the 85 to 110%. What's important about percent recovery is, do you measure it before or after extraction? So I'll leave that one with you guys, but the better people look at it before extraction.
All right, perfect. Well, we've got a question here from Ashley, and she's asking, we are a consulting firm and would like to do research on the potential of pharmaceuticals in wastewater. However, we are having a hard time finding a lab that we can send our wastewater samples to for analysis. Who do you use to analyze your samples?
So we've used several over the years. Some commercial laboratories have developed pretty good methods, like a lot of the bigger national and international ones. But then if you're looking for the really low detection limits and kind of like Ed was saying, the high recoveries, you're often going to a more bespoke outfit. We've worked with the Wet Center out of Temple University. They have a really good program there, and they've actually kind of developed and modified methods over the years to really dial in for particular APIs. So they're good if you're based in the U.S. There are other ones overseas. KWR in the Netherlands is one. But really, the big thing is kind of asking the question of, can they adjust the method based on the matrix that you're dealing with, and get the detection limits that you need. There's probably going to be a bit of cost associated with that method development, though.
All right, perfect. I'm sorry, go right ahead, Ed.
Do you want me to take the next question, or?
Sure, absolutely. How challenging are benchtop evap systems for lab testing?
So we send that out generally. The evaporator vendors will do the testing, the boil down test, and give you a good report. You just got to make sure they're doing it to your expectations. Dave, anything to add on that?
Yeah, and then just making sure that you're working with a good lab to do any analytical testing that you need out of that. If you're just looking at total volume reduction, the bench test lab is usually pretty good. But if you're looking at all the particular compounds, and you want to have that specialized lab.
All right. I'm going to jump to the last question real quick, because it talks about the lab. So the last question there is, what are the one or two main points in wastewater treatment? So this is debatable, but one, if you don't have the analytical methods, you can't measure anything. So you can't do treatability. You can't design a plant. You really got to know the API, and you've got to have the fate and effects data. The second one is if you try to scale anything up in this field of removing trace amounts of material without doing treatability. So analytical methods and treatability are two critical hurdles. And the analytical methods can be a high hurdle. The treatability, you can generally figure out how to resolve that. But those are two significant hurdles that you should be aware of if you're going to embark on this kind of project. And then I see there's a question about antibiotics. What is the most occurring pack group, which I guess should be antibiotics, right? And what is in the antibiotics group? So I would refer you to the USGS 2001 survey of pharmaceuticals and personal care products in the environment. I think they have some graphics and some of the work of Thomas Turns. I wouldn't say antibiotics are the most ubiquitous or pervasive group. I would say your general active ingredients like your acetaminophen, a lot of your blood pressure drugs, and a lot of your antipsychotics are more pervasive than antibiotics. And then we have what temperature contact time is used in thermal treatment for destruction. We haven't used thermal treatment for destruction. We've used it as a concentration process in terms of evaporators. And of course, you got to boil water. Would an existing sewage sludge incinerator achieve the conditions for destruction? So an incinerator would operate at 16, 18, maybe 2200 degrees, and that would definitely destroy anything, including probably PFAS, which is not a pharmaceutical, but is a hard to destroy compound.
Yeah, and with that one, it's really understanding the compound specific properties, but generally you're going to see some level of destruction. And I see Ashley had a follow up about the labs. So it really depends if we'll use a commercial lab. It's really having that conversation with them, I would say, and see what they are able to do, and how well they can develop the method for your specific compounds.
Dave, I also see one by Jack Ambler. It's a good question. It's what about the final solids handling? What types of facilities receive the final solids and not allow them to make it back into the environment? So that's a great question. On the one I just showed you, the treatment plant that we built, those solids went off for incineration, and that's a general policy-driven practice within the pharma industry. Is we don't landfill solids, we incinerate them. Even though on that particular project we did a mass balance, and believe it or not, to my surprise, very little of the APIs were partitioning to the solids. Most of them were being biodegraded, and those that were soluble and slowly or non-biodegradable were oxidized by ozone. So, but I would say if you've got pharmaceuticals in your sludge, you probably need to incinerate it.
And I believe that actually sums up our Q&A for today. This has been great. This was a great presentation. Ed, Dave, we thank you, and we absolutely thank the sponsor for today, Arcadis. This actually concludes today's eShowcase. Thank you all for attending. The recording will be available at webbuyersguide.web.org. We will send all registrants an email tomorrow with that link, and please visit our event calendar to sign up for future webcasts. Thank you all.
Thanks, everybody.
Thank you all.